So this is going to be our EOC review session for day number 2. Today is Saturday, May 7th, and the EOC should be in about 2 weeks. So we got one more session after this. And once again in class, we are going to be going over a lot of stuff too. So this is kind of supplementary, and it's kind of here for you because there's not a ton of you, but whatever. And once again, please excuse any terrible handwriting. It's going to get really bad. All right, so topic number 1, kind of jumping off where I left off on day 1, we're going to talk about the cell membrane. And can somebody just remind me, what's that kind of phrase we use to describe the cell membrane? It's kind of a complicated term.
A jelly-like substance.
That's true. There is a jelliness to it. But it's a certain kind of model. There you go. We use the fluid. It's the fluid mosaic model. The fluid mosaic model kind of looks like this. Here we go. This is going to be really bad. It's drawing time by Mr. Olson.
Oh, I remember that.
Yeah.
The polar and non-polar bodies.
Hey, there we go. Polar, non-polar. There are features of that everywhere here, and there's even some like chunky things in here. There's even some goofy things that kind of stick out. But anyway, we said the cell membrane is composed of mostly these kind of jelly-like things, as we heard. These individual things are called phospholipids. And since there's 2 rows of them, we actually describe this as the phospholipid bilayer. That is a B. Sorry. So this phospholipid bilayer, it does have those non-polar and polar properties that are so important. The outside layer, if I just look at a phospholipid more up close, right? This is what we call their head. And this is what we call their tail. And we actually talked about how their heads are polar, and we said their tails are non-polar, which means the heads love water. They love water, so they interact with it all the time. That's why they face outward, so they can come in contact with water. But the interior layer doesn't like water. That actually means that water has these special pores called aquaporins, which are actually giant protein structures, and that's exactly what this is. That's a protein. And those proteins are surprisingly common in the cell membrane. And that's why we use that phrase, fluid mosaic model, because fluid means it can move around. Mosaic means it's made up of a lot of different parts, and they all can interact with each other and cause all those cool features about the cell membrane. So, and then, of course, there's some like smaller proteins, and they have different purposes. Like you can see, like I put this weird little antenna there. A lot of the times those can be something like a glycoprotein or a carbohydrate that stick on the outside, and they just helped identify certain features about cells. And you'll remember when we were talking about the immune system fairly recently, we said there's like little identifiers on the outside, and there's little receptors to receive those things. A lot of the times they're going to be those glycoproteins. Okay, so we have the basic structure of the cell membrane. Does anybody have any questions on that? Any issues? No? So if you know the structure of the cell membrane, the real concern is what does it do, right? Like how is it actually used in the body? Yeah, so there's always things trying to get inside of our cells, right? And it's not even big stuff like bacteria or viruses. It can be really tiny things like molecules themselves. So this brings us to the idea of transport. That transport we find within cells. Do you remember the 2 different kinds of transport we have?
Active and passive.
Yeah. Passive and active. Now, passive and active differ in a few different ways. Can you tell me what you think passive means?
Without energy.
No energy. And we'll talk more about that energy here in a second, but requires no energy. Active transport, that means it must use energy, right? Right? So we've got no energy, uses energy, but what else is different for passive?
To use the help of, I know one of them uses the help of protein.
You're right. There's one that does use helpful protein. Say again. The triangle. I can draw our best friend. The triangle of love.
Down the triangle,
Hey, would you mind signing in for me? You're here for bio, right?
Yeah.
Okay, cool. So the triangle of love, we talked about him way earlier this year and how he's pretty much your best friend. And if you want to, you can kind of pretend like you can go down that gradient, or you could go up the gradient. If you go down the gradient, that's not going to cost you any energy, right? It's just going to be simply, free. It doesn't cost you anything. So this is going to be passive transport, which means it goes down the concentration gradient. Right? Which means the opposite is true for active transport, which means it's going up the concentration gradient. By the way, I'm Mr. Olson.
Yeah, I know.
Good to meet you.
Me too.
Yeah.
Yeah, I
You
had
could-
Mr. Today, so.
Right. Well, you had. Yeah, I understand. So if you want to take notes, you can take notes. If you just want to sit back, relax, and listen, it's fair game, and you can stay as long or as short as you want to. But it's good to see you. And truly, if you got any questions, just let me know. Okay. So we got passive and active transport. Once again, no energy, goes down the concentration gradient. Active transport uses energy, goes up the concentration gradient. And that's always the thing you can reference. You can always remember your friend, the triangle of love, to help you remember. Now, there's a few different forms of transport. Passive transport comes in three forms, really. We've got diffusion. We've got another one called osmosis. And the other one, I think, Anna, you were thinking about this one. That says facilitated diffusion. It may not look like that's what it says. Well, that's what it says. So those are the three forms of passive transport. Active transport has a few varieties. You'll probably remember endocytosis and exocytosis, right? We even said there's a few versions of this stuff called, actually it's a variation of endocytosis, it's called pinocytosis and phagocytosis. So there you go, two versions of passive and active transport. I should also probably explain, we talked about osmotic pressure. You guys remember by any chance talking about that stuff? So like, let's say we've got cells and they can become hypertonic or isotonic or hypotonic. Yeah, we should probably review that, right?
That's what the water
Yeah, it could change size. So let's go ahead and pick up on that, yeah? 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 Okay. I'm recording this, and that's probably going to be really embarrassing later on. So let's talk about, we've got those different stages. We've got something called hypertonic, something called isotonic, and we've got hypotonic. So hypertonic, isotonic, and hypotonic. I think the easiest one to remember is isotonic because it just means they're balanced, right? Let me draw like a really bad thing here. Hi, Maisie. Go ahead and sign in for me. This is a cup or a beaker. Let's call it a beaker, and it's full of water. And we have a cell in there. For some reason, it's gigantic. And inside of our cell, let's go ahead and pretend we've got some stuff in there, right? That means in my water, I'm going to have an equal concentration of this stuff. And I just want to point out to you, stuff, I'm going to kind of draw that here, stuff in a solution is what we call a solute. Okay? Do you guys remember what the liquid is called? Generally speaking? Yeah, the solvent. All right, so we should have equal concentrations of the solute inside of the cell and inside of the cup. So let's pretend it's salt. You know, like there should be an equal concentration outside of the cell as inside of the cell. And it's a pretty even spacing in my little drawing here, right? I'm surprised it's as good as it seems. In the next one, all right, we've got hypertonic. So if you're hyper, where do your parents tell you to go?
Outside.
Go outside, right? They're all angry at you, which tells us what are we going to find outside? We're going to find the solute. So we're going to see a really high concentration of the solute outside of the cell. There might be a little bit inside of the cell, but not nearly as much. So that means we're going to have a higher concentration. That conc stands for concentration outside of the cell. All right. And so our last one, our poorly drawn beaker with our undifferentiated cell, is going to be hypotonic, which must be the opposite of everything else. So everything's going to be trapped inside of the cell. All the solutes are. Not all of them, but a lot of them. Right? So this means we have a higher concentration inside the cell. Word? I think my beakers look like washing machines. I said machines, by the way. So I think they look like terrible washing machines, but whatever. Whatever. So just to point out to you, this means the solute, the red stuff, how does it want to move in this situation? It wants to move into, right? So as long as it can, it wants to move inside. In this case, isotonic, yeah, it's going to move in and it's going to move out at an equal rate. In this one, the solute wants to move out, right? Because it's got a high concentration, wants to go down the concentration gradient. But we can have cool little situations with water, which, by the way, water is considered the universal solvent. And water, that's all the white space in here. I know I can't really draw the water for you, but how does the water want to move in this situation? Well, there's a higher water concentration inside of the cell and a lower water concentration outside of the cell, which means the water wants to move out of the cell. And depending on how strong the solute concentration is, that'll cause the water to move very quick or very slow. In this case, water will exit very quickly, which will cause the cell to shrivel up, which we call crenation, which explains why your fingers get all pruny whenever you're in the tub. Nobody gets in the tub anymore. Whenever you're in the pool, right? In this case, we're going to have an equal amount of water moving in and moving out of the cell, so that's isotonic's kind of benefit. Everything's just kind of moving in and out freely. In this case, hypotonic, water wants to rush inside. And if that happens, we can actually fill the cell up so much with water that it blows up, which we call lysis. So, have you ever heard of Lysol? I think Lysol is like designed to kill germs. Well, it's probably designed to kill germs because it kind of disrupts the shape of their cell membranes. So, how are we doing? Any questions there? No? All right, moving on. So, we've got cell membrane, we did the diffusion and all that cool stuff. Let's go ahead and talk about cellular energy. Now, this works in a few different forms. There's three main forms of cellular energy. There's photosynthesis, there's cell respiration, and there's fermentation. So let's go ahead and start off with the one we originally started off with, and that's going to be photosynthesis. Okay, so photosynthesis. Do you guys remember, and this is honestly like the most important part of this whole standard, do you remember the formula for photosynthesis? Okay, good. So starting off, we need CO2, that's carbon dioxide. Plants breathe in what we breathe out, so they take in carbon dioxide. They also take in water, because pretty much everything that's alive really needs water, but plants need it for their whole cellular energy process. And of course, they need energy. And this form of energy, by the way, is going to be sunlight. Right? That's where they get their energy from. You know, photosynthetic organisms, not animals, but organisms have their leaves and all that stuff, and they're designed to actually just absorb as much sunlight as possible. And then we have this little arrow here. This little arrow means yields, so that means these three things together will make this stuff. So what are we trying to make in photosynthesis? Oxygen. That's an O2. Oxygen gas, to be precise. And sugar. You can just simply remember it's sugar, or you can remember the generic formula for sugar, which is C6H12O6. And just to point out to you, that's a 1 to 2 to 1 ratio of components in this molecule. If they ever ask you a question on the EOC and they go, oh, this is C8H15O8, that's pretty close to a 1 to 2 to 1 ratio. It just tells you that it's a sugar. That's all it is. But whatever. So that is going to be super duper important. Now, this kind of ties in with the idea of cell respiration.
Which is the same thing.
Exactly. It's the same thing, but totally backwards. So you use cell respiration, which means you take in oxygen, gas, of course. You take in sugar, C6H12O6, and with that, you make carbon dioxide. That's what you breathe out. Water, which seems weird, but that's something you do. And you make energy. Now, when you make energy, you don't make sunlight. That would be kind of crazy. You'd glow and people would stare at you and get blind. But the truth is, we make another form of energy called ATP. Yeah. Very important molecule. It's probably the most important energy molecule we have. There's a lot of other versions out there, but I do want you to understand what ATP is and how that relationship works, and I'll show you on the next slide. But we'll kind of come back to the idea of these energy processes here in a second. But yeah, you will remember when we talked about photosynthesis, we talked about light-dependent and light-independent reactions. We talked about photosystem 2, photosystem 1. Maybe you guys didn't, and that's okay, because you will not see a question like that on the EOC. All right? They're not going to say, like, what stage is the electron transport chain in photosynthesis? They're not going to ask you that, because that's not a requirement for you to know. That's something I taught you because you're in honors, but everybody has to take the same EOC, so it's all built around the regular bio content. All right? So I hope it was challenging, but I kind of warned you a long time ago. It's not on the EOC, so if you don't get it all, that might affect the test grade, but not your final grade necessarily. So let's go ahead and talk about that energy molecule ATP. Does anybody remember what ATP stands for?
Adenosine triphosphate.
Nice. Let's see if I can spell it. That's going to be the tricky thing. I think I did it. I'm pretty sure. Adenosine triphosphate. This actually relates to, kind of relates to a particular nucleotide. It's kind of strange, but We'll kind of get to it if we get to genetics today. So ATP, it actually has a very cyclic relationship. It works in a cycle. We start off with ATP, and ATP is a high energy molecule. That's an explosion of energy. That's what I drew there. And then what happens whenever we want to actually use that energy, we have to lose a little portion of that called a phosphate. Because the structure of ATP is adenosine, which roughly looks like this. Right? Which simply means adenosine triphosphate, tri meaning it has 3 phosphates. And this third phosphate right here wants to break off. So when we force that phosphate on there, it's in its high energy version. And then when we break off the phosphate, we release a bunch of energy that we can use. So when we break off that phosphate, we turn this ATP into something called ADP, which is a low energy molecule, and it stands for adenosine diphosphate. And when we look at the structure of that, it's almost the same, but it's only got those 2 phosphates on there. So of course, if I want to make ATP again, which I do because it's like the major energy molecule, all we simply have to do is add a phosphate. Right? So that's kind of that relationship between ATP, and that's that high energy molecule that matters so much. So when we were talking about, the electron transport chain or the Krebs cycle or the Calvin cycle, what we were talking about is we were talking about how we make so many ATPs or how we break them down to do something else. Yeah. So anybody have any questions on our cute little ATP cycle here? No. No? All right. So let's go ahead and continue back on to the idea of photosynthesis. And I know this seems a little odd in placement, but this is actually how it was designed in the course. When we were talking about photosynthesis, we ended up talking about... Yeah, but I'm going to talk about plant parts. You guys remember talking about the plant parts? I know it seems a little strange to kind of place it in the middle of an energy section, but you probably aren't terribly familiar with plant parts. But we said there's a few things going on, so let's go ahead and describe it. Let's say I've got my, like a tree here. Huh. Yeah, that's what you think it is. That's a tree. Anyway, if this is the ground, what do we have underneath here? Yeah, we've got roots. What's the purpose of the roots? It anchors the plant, but yes, it also absorbs water. And some nutrients. Because there is stuff like nitrogen that's contained in the ground, and the only way we're going to get that into a plant is actually like pulling that up into there. And you guys will probably remember, like, there's the trunk of a tree and branches and leaves and stuff like that. But this is where photosynthesis happens. I should also point out to you that we have another process called transpiration. And so, photosynthesis is basically the idea that plants make sugar, which we just kind of talked about. But transpiration is when gases move in and out of plants, which is a super big deal because, like we showed in our formula for photosynthesis, oxygen is going to be produced by these things, and they have these cool little features that allow them to release that oxygen. So let's go ahead and talk about that process. We have this leaf. Yeah, that's supposed to be a leaf. Deal with it, okay? And we know, like, in our leaf, at the bottom portion here, our cells have these little pockets. Do you guys remember? They kind of look like this. Yeah, so the cells themselves, these two things, these are called guard cells. Right, the whole is actually called the stoma, or if it's plural, it's stomata. And this is where transpiration takes place. Ugh. Right? Also, this is really important because the leaves are really broad on plants because they're designed to absorb more sunlight. That's their job, right? This is why we see competition in nature, because the leaves will grow really broad, and then like trees will compete against other trees. They don't like to grow next to each other. They like to separate so they can stretch themselves out as much as they can. And you've got some pretty unique plants that actually adapt into environments that don't have to compete as much because they get their energy in a little bit more effective way. But yeah, that's kind of a big concept there. You'll also remember we talked about the structures of the sexual structures of plants. So you remember flowers, yeah? Okay, so let's go ahead and hit that up real quick. I know this looks like an elephant, but deal with it. Yeah. Thank you. It's like an elephant with extra tusks. Okay. Yeah, that's my flower. You will probably remember a lot of these parts. We have this portion right here. Let me change colors super quick. We have this portion right here that has two parts. This whole thing together is the male reproductive organ of a flower, or angiosperms is what we're really talking about here. What is that whole male part called?
Stamen.
Yeah. And it's made of two parts. The top part is called the anther. The bottom tubey part is called the filament. And then we've got the female portion, which is the middle portion right there. That is going to be called...
Carpal.
I could take that, yeah, carpal. You can also say pistil. There's a T in there, sorry. I think carpal is more of the accepted term these days, but I could be wrong with that. There's three parts for the female reproductive system. There's the top part called the stigma. There's the middle portion called the style. And there's this bottom portion called the ovary. And if you remember, even very recently, we've been talking about sexual reproduction in animals, and ovaries are kind of generic. That's the term that's used across the board to describe, you know, all of female reproductive organs. Well, the main female sexual reproductive organ. There's also some other little things here. We've got petals. Petals are actually super duper important. They don't just look pretty for nothing. They are actually designed to attract pollinators. We want to make sure that a bee or a butterfly or a bird comes by and like picks up whatever pollen is there and hopefully, you know, makes it do the work of sex basically and spreads those gametes around. There's also these little green things down here. These are called the sepals. And just super quick, we know this process basically works because the anther has a bunch of little tiny granules on it called pollen, and either by wind or by, you know, water or by animals brushing by it or even like just carrying it on their fur, that eventually will transfer to the actual stigma. And the stigma has this tube that runs down the middle and actually connects to the ovary. Eventually that pollen will travel down there and reach the ovary, and then we actually fertilize, you know, essentially the egg, and that'll develop, in this case, into a seed. And then the seed drops off, and then, you know, if it's got water, it's got sunlight, it's going to break open, it's going to like grow, and then hopefully root itself in there and become another future plant. So that's kind of how seeding plants work, which we call angiosperms. There's another thing out there called a cone or conifer, gymnosperms. Yeah, pine cones. They kind of work in a different way because they protect their seeds on the inside. They drop off, they open up, they drop their seeds. And they're kind of designed to just fall from the tree itself. Whereas these guys, they can develop in a few different ways. Seeds, but also fruit. You know, if you've got an apple, which looks like a heart for some reason I drew. The apple is actually, right, if you ever look on the bottom of the apple, that's actually where the anthers were. And the flower would actually be down here. And what you're eating is the swollen ovary of a flower.
Is that why sometimes you could find oranges with like another small orange coming out of the bottom of it?
I've never seen that, but I'll believe you.
Never seen.
Never seen that. Never once. But anyway, that's pretty cool. So, any questions on flower parts, flower structures? No, for sure. All right. Next topic I have is going to be the cell cycle. Oh, no. Of course not. I forgot one thing. There's something out there called aerobic and anaerobic. Whoops, my bad. So, kind of going back to we're on the topic of cellular energy. We're going back to the idea of cell respiration. There's aerobic cell respiration. And there's anaerobic. Aerobic cell respiration is the one that you're already familiar with. We already talked about it, right? We said it requires oxygen. Yeah. It will not function without oxygen. But thankfully, most things that use cell respiration, they are going to be lucky enough to have an alternate energy-making process, which is anaerobic respiration, which an means none. Hey, Donovan, just sign in for me. It's going to be no oxygen, so no O2 or very low O2 is going to be used here. And this is pretty cool because basically what we do is we convert the sugar directly into energy. Now this one makes a lots of ATP, right? But the one down here, the anaerobic respiration, not a lot, very little. I believe aerobic respiration makes 34 through 36 ATP. But anaerobic, I think, makes 2 to 4 ATP. Those numbers might be wrong, but if you double check me, I think I'm right. And we said anaerobic respiration comes in two forms. The first one that you'll find is going to be alcohol. Oh my gosh. I'm getting a phone call. I apologize. Yeah, I'm working, so sorry. We've got alcohol fermentation, and the last one's going to be lactic acid fermentation. So, alcohol fermentation, do you guys remember what the byproduct of alcohol fermentation is?
It's the bubbles in pizza. It's CO2.
CO2, that's true, as well as the tough one, alcohol. Hence the name, alcohol fermentation. Lactic acid fermentation is going to make something called lactic acid, also known as lactate. And lactic acid fermentation is the kind of fermentation we use in our bodies whenever we're using our muscles and we don't have enough oxygen. So if you've been... Yeah, I told you about when I worked out in high school, I couldn't move my arms after working out because I built up so much lactic acid. I was just frozen in pain. It was really embarrassing. And I'll spare you the long details, but I literally couldn't pick up my remote when I got home from school, and I had to eat my Star Crunch, and and I couldn't even move my arms. It was ridiculous. But anyway, lactic acid fermentation, that helps us create a little bit of extra energy while we're using our muscles when we can't breathe fast enough because there's a limitation how fast you can breathe. And then alcohol fermentation, that's going to actually produce basically a 2-carbon sugar called alcohol and a little bit of CO2 gas. This is what bacteria use all the time. Yeast, fungi use these all the time. This is actually what makes bread so delicious, is because you've got little bubbles trapped in your dough, and those bubbles are yeast farts. That's essentially what they are. So, and that's what makes them all nice and fluffy and everything. And this is what makes pizza dough have that unique flavor of yeasty, carbon dioxide smell and taste. And there's also, of course, alcoholic beverages. They are produced this way too. So we've got aerobic, we've got anaerobic. Any questions there? No? All right. So let's actually go ahead and move on to the cell cycle. Cell cycle. There's a lot in the cell cycle. I want to make sure we take our time because there's always questions relating to the cell cycle. It's a big concept. It's a big idea. It's really how you got to be here. So we want to get down to the basis of it. So we've already talked about cells. That was last week. But we do know every cell has their own living method. And that is the cell cycle. So I'm going to draw a circle. It's pretty good. And I'm going to divide the circle. So I'm making a pie chart here, hopefully. Aha, okay. So, the very beginning, I'm going to put a star right here, or an asterisk, or whatever. We're going to start right here on the cell cycle. We start off with one single cell. That's our cell, okay? Starting from here and going onward all the way around is a process called what? Close. That comes next, actually. Interphase. Interphase. So interphase is actually broken up into 3 sections. And I'm going to change colors for you. I don't have many options, but let's go with purple. We've got the first stage, which is called G1. And then the next stage is called
Synthesis.
There you go, which is the S stage. And the last one is going to be G2. Yeah, there is a weird one. Kind of before you get the synthesis phase, we can kind of have the cell kind of circle back and stay in gap 1. We actually call this gap 0. And we do have a few cells that do that. Neurons, brain cells, nerve cells, those don't regenerate very well at all inside of your body. Liver cells are pretty slow to do this. They usually stay in the stage. So if anything happens to them, liver cells can repair, but very, very slowly. But we don't find very much growth in nerve cells whatsoever. So they're kind of permanently stuck in gap 0. Once they're created and they mature, they stay that way because we don't want a whole bunch of our brain cells dying. That would be kind of scary. We like to keep them around as long as possible. So we've got gap 1, synthesis, gap 2. And I'm just going to write those out for you. Gap 1. What happens in gap 1? Growth. The cell overall just gets bigger. And that growth stage is really going to make sure it's set up and prepared. They'll also start to make more organelles too that's trapped inside of them. Then S phase stands for synthesis. Synthesis stage is really important because what happens there?
Replication of DNA.
DNA gets replicated. That's exactly right. And we will try to talk about that today. We'll dive into DNA replication. Pretty important stuff. And then gap 2, which is going to happen with, well, it's going to do what? Mm-hmm.
Growth and check for mistakes.
Yeah, it's going to grow much more. And yeah, this is usually where we do that mistake check. And generally speaking, at these points in between, we usually do check for mistakes. Gap 2 is kind of known for being like the final passing point, the final checkpoint. And this is where we really regulate mutations in cells. And we try to determine, like, yeah, this is a bad cell. We're not going to let this thing make copies of itself. And that usually stops the growth of cancers. So most cancers kind of relate to something weird going on in that period of time. So gap 1, synthesis, gap 2. And then next up, we've got the this section in here. What's that section called? There we go. Yeah, mitosis. Now, I gave you like this goofy... Yes, you're a genius. Thank you, Andrew, for being here. I'll send you a check later. Thank you. What I meant to do is just change the pen color to a darker color. Yeah, so once again, mitosis. I gave you this goofy little thing to help you remember stuff. And it's disgusting, and I know if you didn't have me, you're never going to forget this. Get ready. But you're going to be really proud when you walk up to a family member and you say, I peed on a mat. See? You want to show them that you actually urinated on a floor mat. You're a weird kid, I know. So I stands for interphase. That's terrible. But next up we have PMAT, which actually represents our stages of mitosis. And if you remember PMAT, which is hard to forget now, I know, you've got a prophase, you've got metaphase, you've got anaphase, you've got telophase, or telophase. And then the last one is, fun word to say.
Oh, cytokinesis.
Yeah, cytokinesis. I decided to write that in the hardest way possible, and I'm just proud I got it to fit on the screen. But prophase, metaphase, anaphase, telophase. Prophase, metaphase, anaphase, telophase. There's even like a... There's really like a little secret P in here too. It's called pro-metaphase, and it goes between them. But you're probably not going to get asked that on your EOC, so don't panic about it. But then you'll know it kind of existed. And honestly, it just looks like pro to meta. Like it looks like it's like literally right in the middle between them. It looks like a little cartoon, like you're animating it. So knowing those stages of mitosis. Let's go ahead and let me do my best thing ever. Let me draw it all out because I'm that talented. Here we go. Mitosis. Mitosis starts off with prophase. Now, prophase, we have a cell. Yep, that's a cell. And the cell has just been given the signal, okay, it's time to start dividing. Which actually kind of brings up the idea that the cell is going to slowly change. So we have these things inside of our cells called chromosomes. And we have the thing inside that contains those chromosomes called the nucleus. And at this point in time, the nucleus is going to start to break down. That's why when I started drawing this like dotted line there, right? You're also going to see there's these little things kind of growing outward. Yeah, so we have spindle fibers and we have centrioles. The little circle thingies there are centrioles, and then the spindly things are the spindle fibers. That's what prophase is going to look like. And just kind of to show you this, interphase, the cell has a full nucleus, and like it's not broken up. Those chromosomes aren't loose. They're not separated like that. It's all one big ugly mess called chromatin. But that's what interphase kind of looks like. And then prophase, we go off to this process. Now, I'll go ahead and do it. I'll go ahead and show you prometaphase. And then I'll show you metaphase. So prometaphase looks kind of like this. Let me just kind of space things out real quick. Prometaphase is going to be when these X shapes start to move, right? And these spindly fibers start to move a little bit more as well. That's all that is, just the transition from prophase to metaphase. Metaphase is kind of easier to remember because of this special term called the metaphase plate. You guys remember talking about that? It's when these chromosomes line up in the middle. And by the way, I'm only drawing this with 4 X's, but you really have like a lot more. And I'm not going to bother spending the time to draw them out for you because we have 23 pairs of chromosomes. That's 23 X's. I'm not going to draw that. And they're all like different size, and it's very particular. So it's not just totally random as it might seem here. But these things line up in the middle. This is called the metaphase plate. Right? And I think meta in this case would think about the word, you'd think about the word middle because they're lining up in the middle. And these spindle fibers have grown, and what they're going to do is they're going to attach to the center of the X. Now, just to point out to you, oh my gosh, I'm so talented, so talented. Your chromosomes there, we have something called the I believe that's called the P arm. I think this is called the S arm. And then this middle part is called the centromere. Sorry, I'm breaking that up. But centromere. This is the area that gets attached to with the spindle fiber. And the cool thing is each side of the cell grows the spindle fiber and attaches to that X. And it, of course, looks a lot better than what I'm drawing here. But when that happens, they're locking in place, and they're getting ready to rip these things in half. So you probably remember me telling you about the example where it's like, hey, this is there's 2 giant spiders that live inside of your cells, and they're going to stretch across, and they're going to find the little children in the middle of your cells, and then they're going to rip the children in half. Yeah, I said that. So just kind of imagine these are little children getting ripped in half by giant spiders. Very Hansel and Gretel, you know, very Grimm's fairy tales. Try to think that way. And they get ripped in half. So on the next things that we have anaphase. And anaphase is basically going to be we start to split them. So there's our centrioles. And now we've started to rip those X's in half. So they're like little V's now. And they're kind of getting pulled to opposite sides. We can start to see this middle region right here. It's starting to elongate the cell. And it's going to start stretching apart. We'll even start to see a little bit of caving in right here because it's starting to thin itself out. And then we hit up the next one, which is telophase. We hit it up. We hit it up. And it looks more like this. Kind of like a peanut shape. I'm so talented. Look at my drawing skills, guys. I know. Like, if you need to borrow tissues, guys, to wipe away the tears, there's some over there. But telophase, we start to see the cell start stretching out. The dotted lines that it kind of drew around there, that means each of those now, single chromosomes end up having that nucleus reform around them so they can get protected. And that's what telophase is basically going to be. We start to actually separate the cells. This, by the way, is called the cleavage furrow. And it just means where that cell's going to rip. So it starts to get really thin right there, and eventually it'll just pull until it all rips apart. And actually, a lot of this has to do with the cool protein structures we have inside of our cells, because they actually cause it to rip itself in half, which is a crazy thought. And then finally, we've got cytokinesis. And cytokinesis is going to be, we now have two cells. They have totally broken here. And hopefully they start to look like two normalish cells, because the end of cytokinesis is the beginning of the new cell cycle, which is going to be a new cell, and it's going to multiply. Now, when we talk about mitosis, our goal is to make, we start off with one cell, and our goal is to make two cells, right? But we go from making something called a diploid cell, which has all the genetic information inside of it, to making two diploid cells, right? Because eventually they'll go through synthesis again, and instead of just having one little squiggly, eventually they'll kind of, in a way, start to reform into that X shape. So we'll get that double information. That's what synthesis does. Synthesis stage of the cell cycle. I'm saying every process wrong. So we start off with one diploid and we end up with two diploid cells. Yeah. So that's basically the goal of mitosis. And by the way, I should point out to you that these are identical cells. It's literally your body cloning itself. It's vitally important in that regard. It's got to happen that way. All right. So we've talked about mitosis. Let's go ahead and move on to meiosis. All right, so meiosis, a very, very important process. It is used for not making your body cells, and by the way, mitosis makes body cells, which are called somatic cells in some cases. Meiosis helps you make what kinds of cells?
Sex cells.
Both work, yeah, gametes or sex cells. And the real difference here is that we don't want to make clones of our sex cells. We want to make unique versions of our sex cells. And that's the benefit of meiosis. So just the overarching idea, we start off with one cell, and we turn that into two cells, and then we turn that into four cells. And that should be meiosis. Now, we start off with one diploid cell, and then eventually we start to make four haploid cells. And there's a bunch of cool stuff that happens. So you remember my weird example of you peeing on a mat, right? Well, that process still works out for meiosis. It's almost the same thing, but twice. So we have I for interphase, and then we have prophase, metaphase, anaphase, telophase, and that's just mitosis right there, right? And then we have to repeat the cycle. Now, the cool thing is we don't actually go back to interphase, because that would take a long time for the cell to develop and mature, and they can stay in interphase for a really long period of time. So we just divide the cell, we go through cytokinesis, and then we start right back up in prophase. And then we go back into metaphase, anaphase, telophase, and back into cytokinesis, and then we repeat the cycle. Now, just to show you, like, I know there's a lot of P's and M's and A's and T's. We always try to identify them as prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis, just cytokinesis. Prophase 2, metaphase 2, anaphase 2, telophase 2, and then back to cytokinesis. So how many stages of division are there? 2 stages of division. Now, I'm not going to draw out the whole process again, because honestly it looks like the same thing as mitosis, but there's just a few key differences. So I just want to focus on those differences.
They hug really tight when they switch.
Yep, that's it. So there's there's two key ideas that you got to understand. One is called crossing over, and another one is called independent assortment.
They both happen in Anaphase 1.
Say that one more time.
They both happen in Anaphase 1.
No, that's a 2. That's a 1. So Anaphase 1, Anaphase 2. Is it labeled wrong?
Oh, she was
No,
asking
I was asking,
about.
do those two processes, where do they occur in Anaphase 1?
Oh, when do they occur? Yeah, crossing over is going to happen during prophase 1, and independent assortment is really going to take part in metaphase 1. And that's what I was going to draw out for you in the next slide. So what's actually happening and where. And the stage that they actually take place in is pretty darn important, because that helps you, identify, oh, like that's why these chromosomes are here. That's what's going on. So let's go ahead and draw it out. Yeah? You ready? I hope so. So crossing over. Crossing over is going to happen during prophase, essentially, because what's going to happen is we are going to have chromosomes line up with each other. Now, let me try to draw it out like this. What's going to happen is we're going to get 2 chromosomes that are going to like really just hang out next to each other, like literally hug up on each other. And when this happens, our blue one and our red one will start to do this. Sorry, you have to watch the most amazing artist work ever. They start to exchange pieces, okay? Imagine like you have a family reunion, or you guys get together for Thanksgiving or something like that. And you know how you have that aunt who's just like way too friendly? Yeah, she always comes up to you, she pulls you close, she smells like, old lady. She's got the perfume, she's got the cat hair all over her. She hugs you, oh baby, I missed you, and she kisses you, and then she lets you go, and you've got now cat hair and perfume and old lady makeup all over you. That's going to be crossing over. She's leaving a little bit of herself with you. And that's what happens to the chromosomes. Now, this happens during prophase because those chromosomes are lining up next to each other when they're actually inside of the nucleus, right? So they're hugging each other that way. And then we have, and it kind of happens throughout metaphase 1 too. And then we have, what's that called? Independent assortment. Independent assortment means when we line up our chromosomes in the middle of the body, not the body, the cell body, they can split however they want. So we know we have 2 sets of double chromosomes right here. Well, the blue one could end up going over here, and the red one could end up going over here. But the opposite could be true. The red one could have ended up over here, and the blue one could have ended up over here, right? It's just kind of random chance on which direction they go. Now remember, we have that with multiple chromosomes down here. So that means multiple chromosomes will be able to like move whichever direction they want, which increases genetic diversity. And remember, we said we have 23 pairs of chromosomes. That's a lot of chromosomes. So we're going to basically have like a nearly billions and billions of possibilities for just separating chromosomes. That's not even including the crossing over process, which already makes your chromosomes unique in and of themselves. There's like no way to replicate how a person would ever look like feasibly. Nobody's just randomly going to look just like somebody else based on their genetics. I mean, yeah, they might have some similar features, but genetically speaking, they're never going to be exactly the same, which is a pretty cool thought. So independent assortment, a big idea, major overlying concept. Yeah. So any questions there? I know that's a bit of a lofty idea. I know it's kind of out there, but crossing over, they hug each other and they exchange some genetic info. Independent assortment, they separate however they really want to. They just got to remember one chromosome has to go on one side of the cell and another has to go on the other side of the cell. As long as, they just keep that rule, they can go whichever direction they want. All right? Yeah? Okay. That's pretty much the cell cycle. That's mitosis. That's meiosis. The major points, of course. And if you really don't feel confident in this, like your textbook draws it a little bit better than I can, I'll admit. And you can follow along with the idea a little bit better in there, probably. So let's go ahead and talk about genetics. Genetics, we kind of brings us back to this idea that DNA is really, really big. Like DNA is essential, right? So DNA. We said DNA is standing for what? Do you guys remember? Deoxyribonucleic acid. Good job. And DNA is going to be the genetic molecule found in everything. All living things have a form of DNA. And DNA has a special structure. Do you guys remember what it's called? Shape? Double helix. Yep, that's it. And it roughly looks like this. And there's like these little bridgy things that happen here. Yeah, that's our double helix shape. It's like a ladder that's twisted, if you want to imagine that. Now, DNA has some components, and you'll remember with the last review session we did, and if you remember from class, we said there are things that are called monomers that make up polymers, and DNA is a polymer. So what is its monomer? Its monomer is called a nucleotide. Now, in DNA, there are 4 nucleotides you have to remember. Okay? There's a G, there's a C, there's an A, and there's a T, which stands for guanine, cytosine, adenine, and thymine. Yep. Now, this is how I kind of taught my kids, and I know it's super weird. It's probably the coolest thing ever. But I want you to imagine I know that looks like a pig cow, but deal with it, okay? Oh, crap, you're right. It is a cat. Whoops. I can repair this, no problem. Because it's a cat. That cow sucked anyway, This is G-Cat. G-Cat is the most gangster kitty cat you've ever met. He's so gangster, he wears a gold chain necklace with a giant G-Cat on his chest because he's a gangster kitty cat. And I love the phrasing of G-Cat because G and C and A and T are kind of sticking with each other, okay? And G and C, they pair together, and A and T pair together. So just try to remember, guanine always pairs up with cytosine, A and adenine always pairs up with thymine. And that means, let's say we have a strand of DNA. Let's say we've got a sequence like G, A, T, C, C, T, A, right? That means on the opposite side, called the complementary side, the complementary strand, that means the opposite letter must be attached. So the opposite of G is C. Opposite of A is T. Opposite of A is T. Opposite of T is A. Opposite of C is G. Opposite of C is G. Opposite of T is A. And then opposite of A is T. So we can always find the opposing side of DNA, even if we only read one side. We always know the complementary side. And this is kind of important to remember because basically your body needs to make more DNA from time to time, and that happens during synthesis stage of the cell cycle. And when we do that, all we're doing is we're calling in these nucleotides to pop into place wherever we need them. So I'm apparently really popular today, and everybody that's called me knows I'm working from 10 to 12. How crazy is that, right? So anyway, G to C, A to T, all that stuff. So let's talk about the other version of genetic information called RNA. Now, RNA is a lot like DNA, but it's called ribonucleic acid. Okay, and this is different than DNA in one major way. Well, a few major ways, but it's really the molecule. There's deoxyribo and there's ribose. And basically it means one has no oxygen and one does, which basically means RNA is going to be single-stranded. Okay, it's always going to be temporary. And it's always going to have a change. There's no thymine, but it's been replaced with a U, which is actually a uracil. Okay, so the rules change for RNA. The rules are G pairs to C, A pairs to U. So this is actually where I get to make my drawing from earlier, and it actually would make sense. Those are horns. That's a pig cow. Pig cow is my friend. He's got a shorter tail. Right? That's G cow. He's the most gangster cow, of course, you've ever met. And he's so gangster he can even purposely misspell the word cow. He's that gangster. And believe it or not, they actually get along with each other, both G-cat and G-cow. But you just got to remember those rules. They kind of speak their own language. They get along in a different way. So notice the T is gone, the uracil is there, and this is going to be single-stranded. So most of the time when you see this, it's just a single strand, and it's just got its little nucleotides kind of attached to it. And there's a few different versions of RNA. You'll probably remember these. There's rRNA, there's tRNA, and there's mRNA. And I believe R stands for ribosomal, which actually makes up most of the structure of the ribosome, so it helps it make its stuff, its proteins. tRNA stands for, I believe, transfer RNA. And then last one, and tRNA is going to be used in amino acid sequencing, which is going to help us for protein synthesis. And then we have mRNA, which stands for messenger RNA. So DNA just stays housed in the nucleus. That's its job. It just stays all bottled up. Hopefully it doesn't go anywhere. And that's pretty much it. If we want to move that genetic information to another part of the cell, or actually have it go out and make a protein, because your DNA codes for everything inside of your body. Every single protein you make has to be coded for genetically. We don't want to take our DNA out. That would risk exposure and cause the cell to get messed up. So we just make temporary copies. And that's kind of what RNA's job is. It makes that temporary copy. So let's say the analogy I've used before is like, let's say I want to bake a cake, you know, and I have like a very particular recipe for baking cakes that I love, right? And I'm like, if I ever eat a cake, I want a person to make this cake. So yeah, there's a particular recipe. And so you've got this entire cookbook that has all the world's best recipes in it, and this cake is one of those recipes. Well, if I wanted Haley to make me the cake, I'm not going to hand her the entire cookbook because maybe there's other recipes I want to keep to myself, right? And I'll say, okay, read this recipe, right? And she's like, okay, she's probably going to write down the recipe. But if, she's going to do this, I'm not going to let her walk out with my book. So she's going to write down the recipe on her own piece of paper, and you're probably not going to copy it down word for word. You're probably going to shorthand it as best you can. And it's temporary. You're not going to keep the recipe. So you handwrite your notes about the recipe, and you take that with you, and you go off to your kitchen, and then you start making your cake for me, right? That is going to be what happens in this RNA process. So basically what happens is our information flows in what we call the, oh my gosh, what do we call this again? We call this the central dogma. Of genetics. And that means information starts with DNA, and then it gets transferred to RNA, and eventually that RNA gets transferred into a protein. Okay? So DNA to RNA, RNA to protein. Now, when we get DNA and we just make copies of that DNA, what's that called? Good try. Replication. It's just DNA replication. We just replicate our DNA. That's what happens during synthesis. From DNA to RNA, we have a process called it's one of them. You're right. Transcription. And then from transcription, we move on to a process called translation. So those are those processes that are really important. Now, really, this whole thing is all about designing protein. Like, our goal is to make protein. So we want to understand, the basics of genetics and what's happening in these things moving around and transferring and like changing form. In order to understand how this information is passed on, which is like inheritance, which is Mendel's genetics. So that replication process, let's go ahead and give that a shot. I'm going to give you the most basic version that I possibly can. So DNA replication. DNA replication, of course, we have our strand of DNA, right? And DNA needs to be kind of copied on one side. We only need to focus on one side. And you'll remember there's these 3' to 5' directions. I doubt you'll see anything, but we move from 5' to 3'. That's how we move. So 5' to 3'. It's not a big deal right now. But basically what happens is we've got our DNA. We've got our nucleotides that are kind of there. And what happens is we unzip the DNA. First thing, we unzip. Do you guys remember what that molecule is called that helps us unzip the DNA? It's like a big molecule. It helps unwind that helix shape. That was my hint. It was a pretty good one. Called helicase. Yeah? Anyway, it's kind of like this anchor. It holds the one end of the DNA molecule, and it helps like unravel it and helps open the thing up. And then eventually what happens is we have this molecule called DNA polymerase. And DNA polymerase is really important because it actually kind of pushes it open like a zipper, right? And kind of moves it down the line. And the cool thing is it basically helps us pull in some nucleotides, So there's like free-floating A's and T's and C's and G's, and it helps plug them in place. So what happens, essentially, right, we've got a long line of DNA. And the other half is over here, which we're going to kind of ignore for right now. Okay. What happens is we have the helicase that's moved through here. There's the helicase. We actually come in this with this little molecule that kind of runs down here. It's called a primase. It lays down a primer. So you know how if you're going to paint something, you want to lay down a primer coat. And basically what happens is you put a little bit of paint on the surface, so when you put another coat on top of it, it makes the color appear better, right? It kind of gives you a head start. Well, the primase is really good because what it does is it puts in RNA versions. So let's say we've got a C, a G, a T, and an A. Well, what this does is it still puts in a G, it still puts in a C, it still puts in an A, but instead of a T right here, it puts in a U. Okay, and it just gets the process started. And then what follows is going to be DNA polymerase. And what that does is it continues down the line and it continues to make a long sequence of letters. So let's say it's G, A, T, T, A, C, A. And I know I'm just going really quick here, but then it starts to add the C, the A, the T, the A, the A, the T, the G, the T. And notice it's all only putting down DNA letters. It's only putting in GCAT. And that'll go down that process, until it reaches the end, which is basically a little signal that says stop, which we call the stop codon. Do you guys remember talking about that? Stop codons? Yeah. So we go down this direction, and the cool thing is it works really quick. It's called the leading strand, by the way. Okay. And then the opposite side is called the lagging strand. And what happens is pretty much the same thing, but it goes in an opposite direction. So if this one went this direction, this one's going to go this direction. And this is going to be kind of weird because it only works in sections at a time. Okay, so it'll work this direction. It'll go tuck, 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 tuck. And then it'll stop, and then it's got to go all the way back. Whoop. And then it'll go tuck, 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 tuck. Stop, and then it'll go all the way back, and it'll repeat that process. That's why it takes so long for this process to occur. So both halves have to get copied, but that first half, the leading strand, happens much more quickly. And then after we have, DNA polymerase go through there, we pull another molecule through there to make sure that the primer is getting corrected and it gets back into its DNA form instead of RNA. And then we've made an exact copy of our DNA, and we allow it to let go. It twists back up, and it's just another set of genetic information. I know that's really, really ugly and hard to follow. It's because I'm not using any kind of animated version of this. I would highly recommend, if you want to review this thing and see it in action, go to the school website, and you're going to see, I gave you a couple animations for notes and stuff like that one time. There's a guy, I cannot remember his name right now, but he does a really good job on showing you this process happen. Your textbook, of course, has better instructions than what I can draw out for you, but give it a shot. Try looking through it that way, okay? Okay, so that's DNA replication. So let's go ahead and talk about transcription. Okay, so transcription is going to be the process of basically turning DNA into RNA, and we want to keep it that way. So we're making mRNA, messenger RNA. And transcription is basically going to happen in pretty much the same way that we just talked about, but the rules kind of change, and this is a thing that always throws kids off, okay? So let's say I want to transfer something from DNA to RNA. You got to remember those rules. So let's say I've got a sequence, and let's say it's G, C, A, T, T, A, C. Okay? If I want to make this into RNA, I only need to copy one side because RNA is single-stranded. So if I have a G here, what would I put on the opposite? Like this is RNA. So what's the opposite of G? C. Still true. Still a C. What's the opposite of C? Still a G. What's the opposite of A? U. This is where those exceptions come in, because we said RNA only has uracil. And then T makes a what? A. So that rule still holds true, right? So the only time you'll see anything different is when you're going from A to U. That's the only time that'll show up. So T pairs to A pairs to U, A pairs to U, C pairs to G. And that shows us DNA to RNA transfer is very important. This is essentially transcription, okay? Now, in that transcription process, we make mRNA, but it just wholesale, just copies all of the DNA. But like I was saying to Haley, like if I wanted her to make a recipe for me, she doesn't need to record the whole thing. And there's some bits that you just kind of understand and know that stuff. But our body's kind of smart, and our DNA is sometimes full of junk DNA. So we can actually get rid of some of this junk DNA to shorten it down to make it even better to use. So basically what happens is we have this section of information that's DNA. And then we have the same section, but it now follows the rules of RNA. And then we transfer that into a shortened version. And we only find the things we need. So sometimes there's little sections that we don't need. The sections we don't need, these are called introns. Okay? The parts that we do want are called exons. And then basically we use this molecule called a spliceosome that sticks the exons together and they express what they're supposed to. That's what exon stands for, expression. So we kind of edit it down, you know, like if you're recording a video, unlike this video I'm recording now, you know how you gotta like hit play and you want to say your thing and then you want to hit stop? Nobody wants to see you hit play and hit stop. So you edit those portions out so nobody can see it, right? That's what happens in your DNA as well. So we shorten it down to that short version. And this, by the way, is the official short version of mRNA. Now, if you were in my class, you guys will remember we did the ice cream genes assignment. You guys remember that? Yeah. And that's what that practice was. It was showing you have DNA, you're turning that into RNA, or really mRNA, and then we transfer that into a version called tRNA, which is kind of in relation to rRNA, which basically helps you make the actual proteins. So let's say we're now talking about translation, right? In translation, we get this mRNA. It's just a single thread of genetic info. And we send this to, that's awful. This big molecule called a ribosome. Bear with me, that's really terrible. But the ribosome has two large sections, they're called subunits. And the ribosome is a protein factory, right? So what happens is when we plug in our little section, hey. Bye, Andrew. So when this plugs in here, what happens is we are exposing 3 letters at a time. Okay, and these 3 letters, okay, when they feed in here, they're going to get clamped down on, and this little factory is going to punch in the system, and then we're going to start to see, we're basically going to call out for a signal, and there's this little thing on the outside, and it pulls in these ugly molecules. They're kind of like this shape. and they have a little special attachment on the bottom with 3 little things, and it pulls in these things. And by the way, there's a protein, an amino acid stuck on the outside. And these things come in here and they plug in place. And when they plug in place, what they do is they say, okay, I see the 3-letter sequence I need. So you could have a 3-letter sequence like A, U, G. And then there's that thing called the codon chart. Do you guys remember that thing? It was like a wheel. Yeah, it helps you actually decide what proteins end up there. But there's a wheel version and there's a square chart table version, right? And you got to find those 3 letters, and you find the opposite of AUG. And the opposite of AUG, well, actually, it's already designed to work for you. So you just find AUZ, and you find out this is called a start codon, right? And then you find like CCA, that would be the next one. So we pull in another of these giganto tRNAs that has those 3 letters on it, and it drops off this next amino acid. Then as it like kind of works through the system, what happens is we start growing these amino acids. And the sequence of amino acids is a protein, a polypeptide really. So, you know, maybe this one makes like tyrosine, or I don't know. I don't remember anything in the chart, sorry. There's serine, there's oh my gosh, there's like 20 of them. So lots of different amino acids are going to get formed here. And we just keep putting them in the sequence. Boom, 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 boom. And then eventually we make our protein by dropping off all of these amino acids. Once again, it's very hard for me to draw. It's easier to see an animation of it. I would recommend the Bozeman science videos, the crash course videos. Both of those guys do a wonderful job on showing you the animations. I really particularly like the crash course video because he goes through it very slowly, and like you can see like how it's like a little factory, and it gets to show the buildup of that actual protein. So I'd recommend watching that one. And if you go on the website, I think the video is actually already posted up there. So all you got to do is just click play and watch it. But yeah, translation is basically turning that mRNA into a protein. And once again, if you want practice doing that, you've got your ice cream genes assignment. They're in your folders. Hopefully you didn't throw it away. And you should be able to look at those and go like, oh yeah, I remember how to do this. Now, if you weren't in my class, you probably never did that assignment. And I'll still leave all the information up on the website. That's my school website. So just go to Mount Dora High School website, click my name, and you'll find like under assignments, it should be there. Give it a shot. Look at it. You don't have to like print it out or anything. Just kind of look at it, and you're just trying to translate information there. It's fairly straightforward once you practice it enough, but yeah. So, continuing on with the idea of genetics, and I don't have a whole lot of time. Let's go ahead and try this. There's this dude a long time ago. His name was Gregor Mendel. And Gregor Mendel was the dude who pretty much kind of decided how, units of information seem to transfer. And he kind of has these, he was studying pea plants. Hopefully you kind of studied this stuff, right? And he noticed that pea plants seem to have only 2 color flowers. There's a purple flower and there's a white flower. And he noticed if he took a purebred white flower and a purebred purple flower and bred them together, he only ever saw purple flowers in the offspring. So he kind of used some terminology here, and later on we kind of modified it. But he was like trying to explain what was going on there. So he came up with a term called purebred. But to us, that means homozygous now. And homozygous means same letters. So we have these letters, and they're called alleles. And you can have a big letter or you can have a little letter, right? So if you're, homozygous, you're purebred. He would use the phrase hybrid to describe something that is what we call heterozygous. which means you have different kinds of letters. So that means they could be the same letter, but there's one big one, one small one. So we have a few terms here. We have big A, big A. We have little a, little a. We have big A, little a. Big A stands for homozygous dominant. Little a stands for homozygous recessive. And big A, little a stands for just heterozygous. And we don't have to say it's dominant or recessive because it's got both. So it's not either or. Now, that doesn't mean what it physically appears as. By the way, I should just describe this. These letter combos up here, we call this the genotype. Because those are what the genes look like, right? They're, in this case, you know, the first one here, that's two dominant letters, so that's homozygous dominant. But what do they appear as? That's the question. So if I've got two big letters here, what are they going to look like? Are they going to look dominant or are they going to look recessive? Yes. Right? If we have two little letters here, what's it going to look like? Recessive. And if we've got a big and a little here, what's it going to look like? It's actually going to look dominant. Now, these are the rules for what we call phenotype. Now, there, of course, are exceptions to this, and I'm just going to explain to you. These are the rules of complete dominance. So whenever we have something that's dominant, it totally takes over. So in the case of a heterozygous, it always looks dominant because it just takes one dominant allele, one dominant letter to make it look dominant. So this is the experiment Mendel did. He took two parents, and you guys will remember Punnett squares, I hope. He took two parents, one that was a dominant phenotype, one that was a recessive phenotype, and he crossed them. If you don't remember how to use a Punnett square, just super quick, you take your top letter, you put it in the box, you take your side letter, you put it in the box, right? So this one and this one, right? And the big letter always goes first, you know? It just always has to go first. And the next one, we have a big letter and a little letter. And the next one, we have a big letter and a little letter. And then the last one, we have a big and a little. So all of these kids appear as dominant or recessive? Dominant, right? So all the kids are heterozygous and they appear as dominant. Right? Now, this is important, and this is actually what Mendel did. This was called the P1 generation. And then what he decided to do is he's like, well, let me take only those children and I'll mate them together. Now, this isn't gross because it's just flowers, right? And that's what he did. So he took two heterozygotes and he crossed them together. So in this box, we have big A, big A. In this next box, we have a big A, little a. And this one, we have a big and a little. And this last one, we have a little, little. Right? Now, how many of these kids appear as dominant? 3 out of the 4, right? So we'd actually call that a 3-to-1 ratio. And that's dominant and that's recessive. Okay? Which means 25% of his offspring are going to look, in his case, white, which was the recessive trait. Say again?
The genotype?
This is actually phenotype.
Well, not the phenotype, but the genotype is like heterozygous.
Yes.
Okay.
Yes, ma'am. And then the other 3 kids, that's going to be 75% are going to be purple. Now, he didn't, or you wouldn't use the big letter A. You would use the letter like P or W. But yeah, so that's 75%. So just as a heads up, you know, it doesn't mean that these flowers only had 4 kids. Like, think about humans. Like, they don't always give birth to 4 kids. You know, they don't always give birth to 1 kid. It's just the probabilities that we're measuring here. So when it comes down to this, it just means, like, let's say, you know, Mendel had, like, I don't know, let's say he had 1,000 125 plants that he bred, and he would expect to see 75% of this value to be, dominant. So all he'd have to do is 1125 times 0.75, and whatever that number is how many kids he would expect to look that way. If it's just one kid, that means you just got a 25% chance of having that kid look that way.
What is that equation that we did that we used for this?
That relates to the Hardy-Weinberg stuff.
I remember that.
Yeah, and it's fun. I really enjoyed it. You're not going to see any Hardy-Weinberg on your EOC, so I may not review it. I mean, I might remind you of it, but it's a cool concept. But anyway, how am I doing on time? I got like 9 minutes, so let's see if I can do like a couple forms of inheritance. So we talked about complete dominance. Let's talk about incomplete dominance. Bye, Maisie. Incomplete dominance means it didn't quite finish dominating. So in this case, we have big A, we have big A, and little a, little a. And a lot of the times we just kind of use the same letters. Let's say R is red and W is going to be white. If we get something that's both R and W, so let me try this again.
That would be,
So let's say we got an R, an R, an R, a W, and a W, and a W. RR is going to be what color? Red. What's going to be R and W? Aha. In incomplete dominance, it's a perfect mixture of both. And then in WW, it's going to be white. So if we keep those rules in mind, let's say we mate let's say it's a homozygous red female, and we cross it with a homozygous white male, and that's the symbol for male and female, by the way. How would I set up my Punnett square? What would be my two top boxes be? Homozygous red. Yeah, two big R's, because that's going to be the red, right? And then I've got my homozygous white. I'm going to put them on the bottom over here. W and W. And then I'd cross them up. So I'm going to be having my first box right here, what? R and W. Now, I always put them in alphabetical order. R goes before W in the alphabet, so that's how I put them in there. Yep, it would be true for every single box. And this RW thing means how many of these kids are going to appear as white? 0%. How many are going to appear as red? That is once again 0%, but how many are going to appear as pink? 100%. No choice. It's a guarantee. They will all be pink unless something very strange happens, some kind of mutation. So this goes to show you, once again, homozygous will create what it creates. Now, let's go ahead and set this up, and let's go ahead and cross the heterozygotes. That means we'd get RR, RW, RW, WW. And basically this means what percentage of the kids are going to be white? 25%. What percentage are going to be red? Correct. And we've got pink. 50. So that all adds up to 100%, and we can see those rough ratios. Now, once again, it's a ratio, it's a proportion, it's a percentage. It doesn't show you the exact number of kids that are born. It just shows you the probabilities that you'll see kids born a certain way. So that's going to be incomplete dominance. And yeah, that means this one gets pink, which is going to be a third phenotype. So we get a third appearance, right? That's like a perfect mixture of both. Now, in the next form of inheritance, we have what we call codominance, which means like they cooperate, they get along, so they're both dominant at the same time. So if we're doing red and we're doing white, and we end up with something that's R and W, it's not going to be pink. It's going to be like,
It's kind of like a dog house.
yeah, it'll be like a spotty looking thing. And you've probably seen that on some flowers. They have like two colors at the same time. So that's what we're talking about here. So, yeah, setting up a Punnett square between flowers that are red, and let's go ahead and say in red and white. I'd get RR, RR, RW. Oh my gosh, can't squeeze it in. And that means what percentage of these are going to be white? 0%. What percentage of these are going to be red? 50%. And then we've got how many are going to be the red / white? Should be 50%, right? So it just kind of goes to show you that there are different forms of inheritance and different kinds of things work this way. We talked really in particular about codominance when it came to blood types. And blood types kind of involve both complete dominance and codominance at the same time. Maybe I'll save that for next time. But if you want to go into it, you can just look up. We said there's A-type blood, and there's B-type blood, and then there's AB-type blood. And then we said there's O-type blood, which is going to be little I. But A-type blood can come in two forms. It's either I with an A and a little I. B-type can also be a version where it's IB and then I and then little I. So this would be A-type. This would be B-type. This would be AB-type. And then II is going to be O-type. And so if you cross people with different blood types, it just goes to show you can have a homozygous dominant version. And you can have a heterozygous. And of course, like this is going to be homozygous recessive, so that's still going to be your O-type blood. And I just want to like really quickly just show you with a terrible Punnett square. If we cross somebody that's homozygous A-type blood with somebody that's, I don't know, AB-type blood, you can end up having only kids that will be A-type blood or AB-type blood. And I know the A is just kind of getting away because you could just write A and A and A and B and A and B. But if you go to do a cross and you've got a recessive individual, you're going to need those in place to make sense of it. So in this case down here, you could see there would be a 50% chance that the kid is going to be O-type blood. And then there's a 50% chance the kid is going to be A-type blood because you have the big A and the little I here. And that just simply means the big A still takes over. That's kind of that hint of complete dominance that we talked about. Next time, we will go ahead and we will discuss dihybrid crosses and sex-linked genes, and we'll kind of finish up genetics that way. And then we'll march forward with all the other crazy topics we talked about. Evolution, natural selection, biotechnology, a little bit of that, classification, taxonomy, all those things. Sound good? Sound like a plan? So if you're here, cool. If not, the video will still be posted up on the website. If you want to watch this thing again, you can give it a shot. I know we kind of shotgun through a lot of it very quickly, but I will try to post that up by tonight. So check if you want to watch it again. And if there's somebody you know that wants to make it, like Anna kind of pointed out earlier, like you think a lot of people are watching this already online, cool. Give it a shot. So, yeah. All right, cool. So